Hello friends, very warm welcome to my video. Now today's topic of discussion is photosynthesis in higher plants. We will specifically discuss the light and dark reactions today. Now photosynthesis is comprises of two words which is photo and synthesis. To understand it clearly, the simpler version of this system is to produce food using the photon energy or just transforming the photon energy to its chemical energy. Now, where exactly the photosynthesis happen? That's a pretty good question. And the answer is this organelle which is called the chloroplast. Now we will see the structure of chloroplast. This is the structure of chloroplast, and if we cut through it, we can see these coin-like structures here. These are called thylakoid. Now, if many thylakoids stack together, these are called the stacks of thylakoid or grana. And many more thylakoid stacks are connected through this connecting link. These are called the lumen. And this empty space is called the stroma. Now this whole system is covered by the intermembrane space, and from the outer side is covered by the outer envelope. Now if we specifically cut through or zoom through this section, we can see these are the thylakoid lumen. This is very important because most of the photosystems resides in this place and synthesize the light reaction here. Now two things we have to understand very clearly, that light absorption by plants happen through this antenna chlorophylls. These are also called the antenna pigments, and the most important thing is that chlorophyll A and B plays a major role in absorbing the light. There are also subsidiary pigments such as carotenoid also helps in this program. Absorption spectrum and action spectrum is the basic understanding of light harvesting complex, because in the light harvesting complex lights are absorbed by these specific wavelengths which plants utilizes to continue the cycle. Now the wavelength which plant actually absorb is called the absorption spectrum, and the plants which actually utilizes the absorption spectrum in a particular wavelength are the action spectrum here. So the question is how these energy are getting transferred from one cell to another. To understand it clearly, we have to understand a very basic concept of physics, that is resonance. Now resonance says that energy can be transferred through one particular molecule to another particular molecule without transferring the electron. Only the energy can be transferred. Here the same thinking is applied. Here light comes and this pass through the cells one after another without getting transferred the electron. Only the energy transfers here. Now two terms are important in perspective of NEET here, that one is red drop phenomena and another one is immersion enhancement effect. Now from the early study we came to know that plants synthesize the light in a very particular wavelength which is around 680 nanometer. So if we consider this red line here as different wavelengths, then we can see that in the 680 nanometer there is a peak in the quantum yield here, but there is also a sudden drop here also. It means that plants can synthesize only a particular wavelength of light, and beyond that it cannot capture the light, so the quantum yield decreases randomly. Now the drop is called the red drop. So this is the understanding of that topic, and in regard of that, immersion enhancement effect is also plays a crucial role because it is an important discovery in the photosystem. So how that thought come? When scientists were trying to synthesize the light reaction through the artificial medium, they tried to incorporate different wavelengths into the plant system. And they showed that when two types of wavelengths were given, the photosynthetic rate were higher in a particular position. So they come to the conclusion that there are two different receptors of light which actually captures the light in the plant, and they come to the conclusion of two photosystems where one photosystem captures a particular wavelength of light and another is different from the before one. Now the electron comes from the outer atmosphere to the manganese center, and this transfer the electron to P680, and it is getting and from 680, after excitement, the electron comes to pheophytin. From pheophytin, it transferred to quinone A to quinone B, and after that, cytochrome B6F complex captures the electron. From that part, it comes to the plastocyanin, and from that, P700 got excited. Now the electron come to the photosystem 1. Now the electron transferred to A0 and A1, and then iron sulfur complex to ferridoxin. From ferridoxin, they produce NADPH with using the ferridoxin NADP plus reductase enzyme. Now that whole system actually looks like a jet-shaped structure. That's why it's called the jet scheme of photosynthesis. Now we have to understand the location of PS1 and PS2. We have earlier understood the structure of chloroplast, in which we saw the grana. In the grana there is PS2, and in the stroma and granule edges there are PS1. Now this is the schematic diagram of jet scheme pathway. We can see the transfer of electron procedures. Now this is the visual representation of this two photosystem in the cell. Now this is the schematic of this non-cyclic photophosphorylation photosynthesis. Now there are inhibitors of photosynthesis in the light reaction. There are two inhibitors, chemical compound or inhibitor compound, in which DCMU or dichloromethyluriridine, which actually inhibit the electron transport from quinone A to quinone B, and another one is paraquat, which inhibits the electron transport from P700 excitation phase. Now we have completed the light dependent reaction or light reaction, and now we move on to the light independent reaction or dark reaction. So from the term we say that it is dark reaction, then we don't need the light here, but it's a misconception that it happens in the night, but that's a misconception. It actually does not require light. So the fate of this NADP and ATP what we got in the light cycle will drive the C3 cycle. Now this C3 cycle is also known as the Kelvin-Benson cycle. We all know that. If we simply summarize the C3 cycle, it can be concluded that it is comprises of three simpler steps, which is carboxylation, reduction and regeneration. Carboxylation of RUBP or ribulose 1,5 bisphosphate, reduction of 3-phosphoglycerate, and regeneration of RUBP once again. So if we deep dive into the Kelvin cycle, we can see that ribulose 1,5 bisphosphate, with the help of the enzyme Rubisco and carbon dioxide and H2O, it get transferred to 3-phosphoglycerate. Then with the help of ATP, it transfers into the glycerate 1,3 bisphosphate. Now NADP plays an important role. It reduces it and make the triose phosphates, which are dihydroxyacetone phosphate and glyceraldehyde 3 phosphate. Now we have to remember that these two are isomer of each other. It means they can convert to each other anytime. Now with the cascades of reactions, they make ribulose 5 phosphate, and with the help of the ATP, they once again regenerate the ribulose 1,5 bisphosphate. So regeneration cycle is also get completed. Now Rubisco has dual activity. It means that it has carboxylase as well as oxygenase activity. When the concentration of O2 is high, it actually fixes the O2 and make 2-phosphoglycolate. And 2-phosphoglycolate cannot be used in the C3 cycle to produce the glucose. That's why it has to be gone through the several reactions and make the 3-phosphoglycerate to get into the Kelvin cycle. So plants have developed a system, and this process occur in three different organelles, which are chloroplast, peroxisome and mitochondria. Now the fixed 2-phosphoglycolate transform into the glycolate. Then this glycolate comes to peroxisome, and with the many other reactions it get converted to 3-phosphoglycerate, and ultimately the C3 cycle runs. If we see the overview, we can see that many ATP and NADP are getting used here, but the end result is the same what we got in the C3 cycle. So it is a wasteful process because we are getting the same result but using extra ATP. So it's a wasteful process that we are using extra energy rather getting the energy. So any plant must rescue its operations from that cycle. That's why they have developed a very special mechanism which is called C4 cycle, in which the plants have developed a system and they change their anatomical structure, leaf anatomy, which is called the Kranz anatomy, in which bundle sheath cells are in a ring shaped manner, which are getting surrounded by the mesophyll cells. That's why the CO2 firstly getting converted into the malate in the very beginning where no Rubisco is present. Then the malate comes to the chloroplast where actually the Rubisco is present, and with the help of the NADP malic enzyme it is getting converted to CO2. That's how the CO2 gets into the C3 cycle and the C3 cycle runs. The anatomy, we have to see the anatomy first. Now we can visualize the bundle sheath cells here, and outer side the mesophyll cells are also there, and the darker regions, these are the chloroplast which are present, and ultimately here the CO2 comes. Now C4 cycle divided into almost 2-3 parts depending upon the uses of the enzyme, like NAD plus aspartate type and NADP plus malate type. It means the specific enzyme which converts the 4-carbon compound to CO2 actually decides the name of the process. Another one which is a very important photosynthetic pathway is the CAM. The full form of CAM is the crassulacean acid metabolism. This metabolism process can be only seen in the Crassulaceae family, which are xeric plants. So to get rescue from the high-end heat, they developed a system in which their stomata get opened in the night and they capture the CO2 and makes the malate and they put it into the vacuole, and in the daytime they actually utilize this malate and convert it into the CO2 with the help of the reduction enzymes and make it CO2, actually convert it to CO2 and transfer the CO2 into the C3 cycle. That's how they complete the photosynthesis and ultimately getting the food. So if you see very clearly that the end cycle is same, that C3 cycle and food is the same, but how we utilize the CO2 and how we convert the CO2 is the main thing in the photosynthesis cycle. Thank you.